Now let's continue with the translation, the protein synthesis process. So as we have studied in transcription that DNA forms RNA, for example mRNA is formed, so as what kind of protein to be produced, what are the different sequence of amino acid in that polypeptide chain, or is that protein is now coded in mRNA. So now actually the mRNA with the help of ribosomes and all other RNA forms a protein, and that is what is called as translation. So translation, it is a process in which a sequence of codons of mRNA strand is read or decoded, and accordingly the amino acids are joined to each other to form a polypeptide chain and makes a protein. So translation, here actual synthesis of polypeptide chain with the help of decoding of mRNA. So now these are the requirements for the translation: mRNA, rRNA and tRNA, the ribosomes, amino acid and the suitable energy sources where ATP and GTP is required. Now here this translation is divided into two major steps that depend upon this phenomenon. Like here first step is activation of amino acid and formation of AA-tRNA complex or charging of tRNA. And the second step is formation of polypeptide chain, where actual formation of protein over here. So this formation of polypeptide chain takes place in three steps, that is initiation, elongation and termination. So let's start with the activation of amino acid and formation of AA-tRNA complex. So in activation of amino acid, here it takes place in the first step, and now amino acid are present. Amino acid, where it is present in the amino acid pool in the cytoplasma. So now they should be activated with the help of ATP. Yes, so now this which will act as energy source, and with the help of the enzyme it will help in the activation of amino acid. So for example amino acid is there, so there is for each amino acid there is separate amino acyl tRNA synthetase enzyme. Yes, so as what is going to happen here, this amino acyl tRNA synthetase, we can see here amino acyl tRNA synthetase enzyme. So this is going to react with amino acid. Yes, and ATP will be utilized as a source of energy. So producing this enzyme AA-AMP complex, whereas same time 2-phosphate will be releasing in this process. Yes, and by release of that phosphate there will be the release of energy which will utilize for the formation of the complex called as enzyme AA-AMP complex, amino acyl this complex. So now in such a way amino acids are activated. So here amino acids with the help of ATP and tRNA in presence of amino acyl, this tRNA synthetase amino acyl complex is formed, and this is what in this way amino acid is activated. Now we have learned that tRNA. So how this charging of tRNA takes place? As we know the function of tRNA, it carries specific amino acid at the ribosomes during the protein synthesis. Yes, so as such amino acid should be transferred on the tRNA, that is what it is called as charging of tRNA. So now we can see here this is the tRNA, and as it carry the specific amino acid during the protein synthesis, so when tRNA it carry the, here is the tRNA, here these are the amino acids, when one single tRNA it is carrying this amino acid, so this way we can say charging of tRNA, and that also is happen in presence of amino acyl tRNA synthetase enzyme. So now such enzyme AA-AMP complex is going to react with specific tRNA depend on the anticodon or codon we have learned, producing AA-tRNA complex, and enzyme is going to utilize again. So now in amino acid tRNA complex, as there are two arm, yes, two arm, that is 5' arm and the third primer arm. So what happened here? Third primer arm and 5' arm, in that the third prime arm there is a site for loading of amino acid. Suppose this is the tRNA complex, this is the tRNA, yes, so this is the tRNA, and here the third prime arm, we can see this is the, yes, this is the 5' and this is the third prime. Yes, so at the third prime here amino acid is loaded, this third point. So we can say the third prime arm, there is a site for loading of amino acid. So as amino acid with its carboxylic group is going to react with hydroxyl group from last nucleotide of tRNA, that is always A, and there will be formation of AA tRNA complex in that way. So in this way thousands of amino acids are charged. Here whatever amino acids are present in the amino acid pool, so this way the thousands of amino acids they are going to charge, and tRNA also charge. So as now each tRNA is going to have one amino acid. Each tRNA here it will react with this, and here one amino acid they are going to have. So now this tRNA will depend on the codon anticodon matching. So it will be carrying that amino acid at the site of protein synthesis, that is the ribosomes. So when they are going to carry these all tRNA, then what will happen here, they will reach to the site of protein synthesis where it is going to takes place at the ribosomes. So this is about the first part, that is the activation of amino acid and charging of tRNA complex. Now coming to the second part, that is the formation of polypeptide chain. So now here how this polypeptide chain is formed, that is by three different steps. That is, first is initiation, then after initiation elongation, and then termination. So for initiation, here it requires the initiation codon, that is A, U and G, that we know this is the initiation codon, and it requires ribosomes. Then what it requires? It requires amino acid, yes, which are present on the tRNA, and some initiation factor. So it begins with the formation of the initiation complex, which require the mRNA having codon for a polypeptide, and here ribosomes, that is smaller 30S and the larger 50S subunits of ribosomes, and initially here AA1, that is amino acid 1 with tRNA, the first amino acid which will come make the complex with the tRNA, that is amino acid 1, indicate as AA1, and ATP and GTP as the source of energy. The process starts with the binding of mRNA on the smaller this ribosomal unit, that is 30S, and then here AUG, which is present where on the mRNA, which initiate the process of protein synthesis. So here initiator tRNA that binds with the initiation codon always, that is AUG, by its anticodon, that is UAC. So through how it is going to bind? Through the hydrogen bond, and it carries activated amino acid methionine in eukaryotes or formylmethionine, that is FMET, in prokaryotes. So here now in case of prokaryotes there are only three, we can say, which has, whereas in eukaryotes there are 6 to 7 initiation factor. In prokaryotes 3 initiation factor. Eukaryotes 6 to more than 6 initiation factor is there. Whereas for termination there are termination codon, that is 3 of them, as we know here, UAA, UAG and UGA. And for each there will be one terminator factor, any one. So out of these 3. So let us check now how initiation is taking place. Yes, so what is happening in elongation and then what is happening in termination. So let's see. So for that we will make one diagram. So we can see ki how, suppose this is the mRNA. Yes. So this is what? One mRNA. And now the initiation. For the initiation, here initiation codon is there, that is AUG. Yes, here smaller subunit is going to join. Yes, and here the larger subunit of ribosomes will join. Yes, and here many of the tRNA is there, which is going to carry the amino acids from the amino acid pool. So here suppose this is the tRNA, it will carry this amino acid from the amino acid pools, and here 3 sites are there on this ribosome. Here 3 sites are there. First one is here A site. Yes, then here P site and here E site. So here 3 sites will be there. So now in the initiation, what is going to happen, that mRNA which is present on 5' and 3' end having the initiation codon. Yes, so this is the 5' end and this is the 3' end. So 5' end always have the initiation codon, that is AUG, and in very rare case it is GUG. Yes. So mRNA gets attached with the smaller subunit of ribosomes, or ribosome is going to attach with the mRNA. So says 40S in case of eukaryotes and 30S in case of prokaryotes. So we will generalize the statement as smaller subunit. So as smaller subunit gets attached with the mRNA, we can see here this is mRNA and this is the smaller subunit, here brown part. So that gets attached. So now with the smaller subunit, it will attach in such a fashion like the smaller unit is going to have 3 sites. That is first is A site and the second is P site, and next one is, you can see here, A site, P site and E site, that is called as exit site. So ribosomes have these 3 sites. A site is named as amino acyl because here at this side amino acids comes and joined at this side. Then here next is this peptidal site, as these amino acids are going to join at the P side. When they translocate from A to P, they are going to join with the peptide bond, so that it is peptidal site. And next one is exit site, where tRNA is going to exit after leaving its amino acid. So as here mRNA gets attached to the smaller subunit of ribosomes in such a fashion, so as this initiation codon AUG gets attached or come on P site. Yes, whereas we can say as a rule a second codon will be going on the A site always. And now this is the only exceptional case that AUG initiation codon which goes directly on the P site. So rest all other, including the terminal codon, all of them they will be coming on the A site first and will be shifted to the P site. So firstly they will come where on the A site and then shifted where on the P site. So AUG, as we know, initiation codon, so will attract the complementary anticodon which is UAC. Yes, so AUG is the initiation codon and the anticodon of AUG is UAC. Now which is present where on the tRNA, which is present on the tRNA. So as temporary bond like hydrogen bond will be taking place in a complementary basis of codon and anticodon. Whereas now this tRNA is going to have first amino acid. Yes, and now it will be always methionine in case of AUG. So always the tRNA is going to have, the first tRNA is going to have first amino acid, and that is always methionine. So that is in case of AUG. So in eukaryotes it is regular methionine, whereas in prokaryotes it is a formulated methionine. So now we can say one such complex is produced. Yes, so what is the name of this complex? That is a smaller subunit of ribosome. Yes, this complex is produced. So what is there here? You can see here the smaller subunit of ribosomes. Then this is the we can say this is mRNA. Yes, then here amino acid and tRNA. So this is the what we can say MET, methionine. So this is what the one complex is produced. Now thus the complex is produced, and now what is going to happen here? A larger subunit. You can see now one complex is produced here. This is the initiator tRNA. Here one methionine. Yes, so this is produced. Now what will happen after forming this complex? A large subunit of ribosomes gets attached with this complex. You can see here. Here in this case, here large subunit of ribosomes gets attached with this complex and forming an entire ribosome. So this is what is called as initiation. So this is what the entire complex is, what the initiation. For this initiation, factors are also essential, initiation factor. So after this, here initiation, it comes to elongation. So here during the initiation, this complex is going to form where this smaller subunit of ribosomes and larger subunit of ribosomes, they gets attached where on the mRNA along with that tRNA. It makes the complex with the amino acid, just charged tRNA, they will also comes and attached with it. So this is what the complex is formed. Now after that, here elongation. So now what happened here in the elongation? Here we can see in this, here an empty tRNA leaves from the E site. See, if it will come where? Firstly it will come at the A site. Then suppose here is the A site, here is the P site, and here is the E site. So firstly it will come at the A site, tRNA. Then from the A site it will reach to the peptide bond site, that is P site, and it is leaving its amino acid at the P site, and then this tRNA is leaving from the E site, that is the exit site. So it leaves from the E sites, and only the amino acid tRNA complex bind at the P site directly, while all the other incoming tRNA complex gets attached first at A site and then shifted to the P site and then exit from the E site. So now comes to the elongation. Yes, so during this elongation, what is going to happen here during this process? So you can see during this process, the activated amino acids are added one by one to the first amino acid, that is methionine. Yes, so that is here it indicates by, we can say A1. Yes, suppose this is the methionine. Now this amino acid binds with the amino acid binding site of tRNA, and this results in the formation of tRNA amino acid complex. Suppose if it is attached with the tRNA, so then it forms the tRNA amino acid complex. Suppose this is what the tRNA, and when it attached with the tRNA, then it forms the amino acid tRNA complex. So here with this side it is going to attach. So now what is happening here? So situation now on the P side, the first codon and the first anticodon, yes, first codon and first anticodon mRNA, first you can say amino acid is present. Now what is going to happen on A site? Yes, so firstly codon recognition. Here amino acyl tRNA molecules, they enter the ribosomes where at A site. Yes, tRNA with amino acid. We can see this is the larger ribosome unit, and this is the smaller subunit of ribosome. Yes, and here we can see here's the P site, is the A site. Yes, so what is going to happen here? and after P site, this is P site, and here will come the exit site. Yes, so what is going to happen here? This anticodon binds with the codon by the hydrogen bond. Then amino acid on the first initiator tRNA at P site and amino acid on tRNA at A site, they join by the peptide bond. So at this time, the first tRNA at P site is kicked off. Yes, as it moves to the E site, from the P site to E site, and then it kicked off, and the amino acid with that tRNA is going to attached at the P site. Yes, tRNA which is present at the P site, it will attach with that with the peptide bond. So that's why P site is called as peptide bond site. So in this situation, we can say the P site, as we know, the first codon, first anticodon, mRNA, first amino acid, everything is first in the situation, elongation. Now what is going to happen here? A site, there is the second codon, consider as GGG, suppose. So as it will be attracting a complementary anticodon containing tRNA, which is CCC. Suppose the codon is GGG, so it will attract the complementary anticodon always for the GGG, it will be CCC, and this CCC is now going to have a second amino acid. Yes, we can see here CCC and here GGG. So that is what is going to have the second amino acid. Now what is going to happen? That is the codon and anticodon. In case of second, that is A site will get complementary pair with each other. And now what is going to happen? That is the amino acid AA1, or we can say the first amino acid and the second amino acid, they will react with each other. Yes, of the first amino acid, the AA1, suppose first amino acid, the carboxylic group, that is going to react with the second amino acid with its amino group. So very specifically it happened, this for means whatever the amino acid is present on the P site is going to react with this carboxylic group, whereas whatever amino acid is present on tRNA which is on the A site is going to react with the amino group, and then there will be formation of a peptide bond between the two amino acid. Yes, we can see here one amino acid, yes, methionine, is reacting with the proline. Yes, and then here many more amino acid will come and join with each other by the peptide bond. So in this way here, amino acid one and just making a bond with the other AA2, yes, with this amino acid. So this is what the very important part, where it is going to react with the one amino acid is going to form a peptide bond with the other. So in between two amino acid, here peptide bond is going to form. So now it is very important, the same time now for this it requires GTP. Yes, that is guanosine triphosphate as a source of energy, whereas same time what is going to happen, there is a breaking of the first amino acid bond with the tRNA. Yes, and that tRNA will move where at the exit side then. So same time what is going to happen, we can see, suppose this is the tRNA. If I am saying this is the tRNA, so what happened? So in one case I am saying it is attached with the tRNA, this is the amino acid, and then what is happened here? Here breaking of this, suppose this is the tRNA, here breaking of this. Yes, so amino acid breaks with that. So same time what is going to happen here? Amino acid is going to shift from 5' end to 3' end. We can see here this is the 5' end, this is the 3' end, and here you can see amino acid, it is shifting. Yes, shifting with respect to one codon, which is and this process is what called as translocation. Yes, as this ribosome is going to shift, this is the ribosome entire, you can see here it is shifting, you can see it is shifting, here you can see shifting. So here this is what process called as elongation. Yes, So this elongation is continuous as it reaches to the stop codon. So as now because of this translocation, that shifting of ribosomes from 5' end to 3' end. Yes. So here this is the 5' end, this is the 3' end, continues the shifting of ribosome from 5 to 3' end. This is the stop codon. Then what will happen here with respect to one codon? What is going to happen now? We can see AUG, which was there on the P side, is going to come on the E side. And now that the second codon which was on A side says GGG. Yes, we can see here second codon, that is GGG, or we can say is going to come on the P side, and then the third codon which is now going to come on the A side. So A site, if A site will be free, adenine site, so then the next codon will come on that site. Now simultaneously what is happening, the tRNA which was carrying first amino acid, yes, is empty now. So it will be released from AUG and it is going to exit now. So that is why this site is called as exit site. Whereas now on P site there is a presence of tRNA which is carrying a dipeptide having amino acid 2 and amino acid 1. You can see this is amino acid 1, this is amino acid 2, and here tRNA is going out from the exit side, and then the other tRNA, it is just coming with the other amino acid. So in this way, and again if it is adding from here, then this tRNA is going to exit from the exit side, and in this way here the process continues and chain elongates. Yes, so what is going to happen here? So in this way there is the carrying of dipeptide having AA2, AA1, and then the same thing is going to happen, and that is what called as elongation. Just amino acid 1, AA1, suppose this is the AA1, yes, it is going to make bond with the AA2, yes, this is what, then AA3. So in this way they are going to make a chain like structure. Yes, so this is what called as elongation, and that all elongation happens at which site? That is always on the peptide bond side, that is the P site. So same story is going to repeat on the third tRNA, then it is going to come and then it is going to have third amino acid, that is, as I told you here, AA3. Yes, so codon-anticodon reaction will take place. Then now on the P side, what we have suggested that on the P side amino acid carboxylic group will be reacting with the amino acid having this amino group on the A side, and once again a peptide bond will be formed, that AA2, that is amino acid 2, will be released from its tRNA and the process is going to repeat, and this is what the elongation. So as likewise every time translocation will occur, every time when one amino acid is going to come at the P site, then the ribosome will shift towards the termination end, towards the 3' end, and in this way the translocation also takes place and the chain also elongates. So as likewise every time translocation will occur, and for translocation it requires GTP as a source of energy. Now likewise each and every codon will be carefully read, and every time one amino acid will be re-added and the chain of a polypeptide will keep on getting elongated. That is why the name of this step is given to as elongation. Yes. So now we understand the elongation, and the side by simultaneous the translocation also takes place. So the tRNA at A site carrying a dipeptide at A site that moves to the P site, and this process is what? Translocation. So in translocation, both the subunits of ribosomes, they move along in relation to tRNA and mRNA. So hence this tRNA carrying dipeptide and now gets positioned at P site of ribosomes, making A site vacant. Yes, at this site the next charged tRNA molecule carrying amino acid will be received, and now during this process first uncharged tRNA is discharged from the E side, and as the tRNA, how it gets discharged? When it leaves its amino acid here, then what will happen? If it is attached with the amino acid, then it is charged, and as it is releasing its amino acid at the P site, and then as it reaches to E site, as it has no amino acid, then we can say this tRNA is uncharged. As it is uncharged, then it is going to discharge from this ribosome from the E side, that is from the exit side, and this process is repeated as the amino acids are added to a polypeptide. So it takes less than 0.1 second for the formation of polypeptide bond. Here you can see the diagram. Now likewise it will keep on in up till the termination codon will coming in the picture. Now let us check what is going to happen during this termination where the release of polypeptide takes place. So now what is going to happen here? Every time, because of translocation, ribosomes has come in such a fashion so as on the A site now termination codon has placed. Yes, termination codon where it placed on the A site, whereas on the P site there will be a tRNA which will be carrying a polypeptide chain. So polypeptide chain always at the P site. Yes, so as polypeptide chain is present on the tRNA which is present on the P site, that tRNA is present where? On the mRNA, and such codon is present where? On the P site. Yes, so whereas now at the A site there is the presence of terminator codon. Yes, now already we have learned that in the termination codon there is no tRNA. Yes, at the termination point. So what happened here? So as now on A site there is a termination codon. As it is after this translocation, as the ribosome, this reaches towards the termination end. So as instead of tRNA, what will happen here? Now each termination codon is having its own specific termination factor. You can see here, like at the end mRNA there is the stop codon. These three are the stop codon. Out of these three, any one will be there at the termination point. It is exposed where? At the A site, and it is not read now, and joined by the anticodon of any tRNA, and the release factor binds to the stop codon, then thereby terminating the translation process, and the polypeptide now released in the cytoplasma. Yes, so here after this attachment of this termination factor or the releasing factor on the A site, what is happened? Once again this translocation will occur on the ribosome, and now what is the sequence of translocation? That shifting of polypeptide chain, yes, to next tRNA. So now since there is no tRNA present over here at the termination end, there will be a release of polypeptide chain. Why release? As there is no tRNA present over here at the termination end. So no polypeptide chain is formed there. So what will happen here? Then whatever chain is formed, that is going to release at that point. So now two subunits of ribosomes that also dissociate, and last tRNA that will set free in the cytoplasma. So finally mRNA is also released in the cytoplasma, and it gets denatured by the nucleases immediately, hence mRNA is what? The short-lived here. So in this way, here polypeptide chain is formed. Yes. So that is what after the dissociation, as this smaller subunit of ribosome, larger subunit of ribosomes, so they all will get dissociate with each other, and that is what is called as termination. So as we are going to get a polypeptide chain, we can see here, this is the polypeptide chain. You can see here in this picture, you can see dislocation of this is the smaller subunit, this is the larger subunit, this is the tRNA at the termination point, no amino acid is going to add, so that here this polypeptide chain is again getting free with that, so everything is getting free here. mRNA also just released in the cytoplasma, so everything it gets denatured in the nucleases immediately, and this mRNA is, we can say it is short-lived. So now this is about the termination. Now what is going to happen every time because of translocation, ribosome has come in such a fashion so as on the A side now termination codon has placed. Yes. So whereas on the P side there will be a tRNA. Yes, which will be carrying a polypeptide chain. So as such polypeptide chain is present on the tRNA, that tRNA is present on a mRNA, and such codon is present at the P side, whereas now at the A side there is presence of termination codon. Now already we have learned that this is in you can say termination codon, there is we can say there is no tRNA. Yes, so as now on the A side there is a termination codon, so as instead of tRNA, what will happen? Now each termination codon is having its own specific termination factor. Now matching termination factor will be getting present present on A side, and now after attachment of this termination factor or releasing factor on the A side, what will happen? Once again the translocation will occur on the ribosomes, and now what is the sequence of translocation? That is shifting of polypeptide chain to next tRNA. Now since there is no tRNA present over here, there will be a release of polypeptide chain. So as we are going to get a polypeptide chain, and both the ribosome units are separated. So that is about the termination. So this is about the translation which we have studied here. Now how one polypeptide chain is produced, and that polypeptide chain may act directly as a protein, or likewise many polypeptides will be coming together to make protein. It depends on the nature of the protein. Now what is happening, that every time ribosome is starting with AUG, keeps on reading elongation and termination and releasing the protein or polypeptide molecules. So now likewise several number of polypeptides or likewise several number of protein molecules required, and they can act as structural protein or functional protein. So this is about the translation, about the second part of central dogma. So now we have finished with the transcription and then translation. Now in short, just for revision, I want to show you one 3D animation video so that you can understand in the animated form. If you will see this video, you will understand better about the transcription and translation. So let's watch. In order for our body to function, we need to supply them with a variety of nutrients we get from our diet. Our body cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our body cell can absorb. And where are the instructions to manufacture these and all the different types of protein we need to stay alive? So the instruction to make protein are contained in our DNA. DNA contains gene. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes can contain regulatory sequence that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus where DNA is used as template to make messenger RNA. Therein, translation, which occur in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make messenger RNA strand with the help of enzyme RNA polymerase. This process occurs in the three stages: initiation, elongation and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled by either permitting or blocking access site by the RNA polymerase. Binding causes DNA double helix to unwind and open. Then during elongation, the RNA polymerase slide along the template DNA strand. As a complementary base pair up, the RNA polymerase link nucleotide to the three end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript to complete and the RNA polymerase, the DNA strand and the messenger RNA transcript dissociate with each other. The strand of messenger RNA that is made during transcription includes region called exons that code for protein and non-coding section called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed and modifications such as 5-cap and 3-poly-A tail are added. This process is called intron splicing and is performed by a complex made up of protein and RNA called as spliceosome. This complex removes the intron segment and join the adjacent exon to produce a mature messenger RNA strand that can leave the nucleus through nuclear pore and enters the cytoplasm to begin translocation. How is this information in the mature messenger RNA strand translated into a protein? So, the nitrogenous base are grouped into 3 letter codes, that is codons. The genetic code includes 64 codons. Most codons code for specific amino acids. These are 4 special codons, one that codes for start and 3 other codes for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occur between the code of the messenger RNA and anticodon of the tRNA. After the initiator tRNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called E, P and A site. During elongation, individual amino acids are brought to the mRNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a tRNA molecule corresponds to a particular amino acid. A charge transfer RNA molecule binds to the A site and a peptide bond forms between its amino acid and then one attached to the transfer RNA at the P site. The complex slides down one codon to the right where the now uncharged tRNA exists from the E site. and the A site is open to accept the next tRNA molecule. Elongation will continue until a stop codon is reached. A released factor binds to the A site at the stop codon and polypeptide is released from the tRNA. In the P site, the entire complex dissociate and can resemble to begin the process again at initiation. The purpose of translation is to produce polypeptide quickly and accurately. After dissociation, the polypeptides may need to be modified before it is ready to function. So, modification takes place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestine, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi and then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for the most physiological function of the body to occur properly, such as breaking down of food particles in digestion, and the process of transcription and translation make the production of protein possible. So in this way, we had studied in detail about transcription and translation. So this is what the central dogma of protein synthesis.